Okay. So we got up through this slide before. We were talking about plant pathology, so plants suffering, being attacked in their world. So as I mentioned before, there's potentially jobs in plant pathology. Always room for someone to hire you to tell them what's wrong with their plants. So knowing the differences among the organisms that are on there, the relative kinds of harm that they cause, where to look for them, and so on. These are important skills if you want to get into plant pathology. I don't know any plant pathologists personally, but it sounds like a pretty fun job, I would think. So we got through, these are relatives of aphids. We started off with aphids and the idea that they are basically suction feeders, so they pierce the plant and suck out its juicy parts. And related to aphids are leaf hoppers here, scale insects. Sorry, jumped around too much. Scale insects, mealybugs, whiteflies. If you ever want to meet these organisms, we have these in abundance in the greenhouse. Be happy to show you our greenhouse pests. So I don't want to feel like we're too far behind. Try to get through the rest of the content here and then move us on to the scheduled content for today. So moving underground, nematodes are pretty abundant in soil, and some of those are pathogenic nematodes. They'll attack living parts of plants and sometimes actually cause some alteration to the root structure by basically kind of cysting up or burrowing into roots and living in there. So these are, if you did genetics class, this is an organism we look at in genetics, a type of organism called the roundworm or nematode. They're related to insects and very abundant in the world and sometimes attacking plant roots. So as with a lot of the plant pathology content, there's, I think really knowing that you've got this organism as your pest would come down to finding these actually in your soil, in the roots, maybe even like a DNA evidence of them being there. As far as the phenotype of what they show, the plants, of course, just unhealthy above the surface and dying below ground, maybe some evidence like these root knots that are induced by this type of nematode here. So nematodes, fungus, we've covered fungi, some of these fungi. A lot of them are just like fungi in general. A lot of them are decomposers of things that are already dead. Some fungi attack living plants, and here's some examples. So just, you might have encountered powdery mildew. This is something that shows up on the surface of leaves, gives it a kind of a dusty white color to it. Doesn't seem to have a terribly detrimental effect on the organism, but it can pretty well cover the plant. Powdery mildew. This is one in the upper right. This black knot fungus is something we see on our cherry trees in the area. So like black cherry and relatives. It's a fungus and it causes this kind of swollen, sort of burnt look on the branches. And then a little more exotic kind of fungus. This one has a look a little bit like a gall that we saw. The galls were induced by insects. This is one that is the fruiting part of a fungus that's living inside of this white cedar plant. So characteristic of the species and not the best thing for that plant to have on it. So there are fungi that attack plants. They can be fairly specific, and some of them are drawing attention for being pests of our crops. So around the world, we have a lot of plants that are grown in many places. And in a lot of cases, they're like the same species and sometimes very, very narrow genetic versions of those species. So if you breed a plant over and over again, you get basically like a genetically homogeneous form of this plant, and it gets passed on throughout the world. So coffee, this is a plant that's native to Eastern Africa and now is found all throughout the world, mixing up organisms with different pathologies in different parts of the world. Sometimes these show up as new diseases. So this is one, it's called a fungus here that attacks the leaves, as you can see, of the coffee plant. every once in a while you see one of these headlines. I don't know if you've encountered them. Like, is this the death of bananas? Right? Are we going to lose coffee forever? Certainly a concern, right? So if we have genetically nearly identical coffee plants around the world, and there's one fungus that can attack them, destroy them, ruin the crops, maybe that one fungus, if it's spread throughout the world, could in fact take out all the world's coffee. What do you think? Kennedy says no. That's a very, very firm answer. You think this is not the end of coffee? It's a bold answer. Slim chances. All right. Well, I appreciate your skepticism. I think I share some skepticism there. Multiple species, that's a good point. So our coffee plants, yeah, two big species are cultivated. There's the Arabica coffee that you've probably heard of, touted as one of the more, it's the more expensive, the fancier version. And then there's one called Canephora or Robusta, which is more, not quite as high end, I guess. Shows up in your more like store brand coffees. So that's a good point. I don't know which species this fungus is more of a problem on, or what parts of the world. I'd need to follow up on that. But it's possible. So the way that we grow crops actually kind of encourages this kind of problem. So if you grow the same crop plant densely together in big plantations, you're setting up an environment where if a fungus gets in there, it's got a full food source, right? So we have ourselves to blame for some of this. We're putting crop plants out of their natural conditions and into these artificial conditions that sometimes are really good for getting pathogens around. Fungus in particular, if you think about how fungi get around, right? So they have microscopic spores, pretty hard to prevent them from getting around. They get around in the air. And so if people are moving from place to place, if you're a person who's maybe doing consulting on coffee plantations or something, you could be passing this around between coffee plantations and not know it. So something to think about. Maybe, the next generation might see a big change in what happens to our global coffee supply or chocolate supply. Maybe you weren't affected by coffee, but you're more interested in the fate of chocolate plants. So here's another one. Same thing. Another one of these headlines comes through like, is this the end of chocolate? And it's, again, a fungus that's specific to chocolate, takes advantage of the density of chocolate plants grown on plantations. And of course, big money in all of these crops, big money in coffee, chocolate. So lots of money, lots of science being put at preventing these things from getting around and destroying the world's plants. So maybe keep this in mind. We'll bring this back up as a topic. We talk about genetic engineering as our last topic of the semester, I think, one of the last two. Maybe new ways to get around your coffee problem. I guess maybe kind of related to this, if you want to, we're talking about kind of luxury foods, right? Chocolate, coffee. I would really feel sad if I didn't have my coffee, but I would not be, my health would not be affected drastically. My survival, certainly. However, this was a situation you've probably heard of that people's survival was most definitely impacted. This is a pathogen that affected potatoes. And the potato story is an interesting one. Potatoes are native to South America, and they were introduced to the world when people were exploring in their ships and sailing around the world, discovering potatoes for the first time and discovering that they could be grown lots of places. And in Ireland in particular, of course, they took over as a very popular crop. So this really kind of saved Ireland from a certain amount of famine when potatoes were introduced. This was a crop that could be grown with not a lot of it doesn't take a lot of care to take care of your potatoes. And in colder parts of the world, when potatoes are done growing, their tubers stay underground, and so you can sort of harvest these things year round. A nice crop to have around. And of course, that's why they were adopted by the people who were native to South America. So Inca and their descendants are the people who were cultivating potatoes before Europeans found them. But then the problem arose. This is a pathogen of potatoes. So potatoes had been adopted as a really popular, very effective food supply for people, and they had come to be largely dependent on potatoes for their food. So in comes this pathogen called Phytophthora, which is a kind of a relative of fungus called an oomycete, and just destroyed the potato supply. And major world changer here at this point. So biggest effects in Ireland, the so-called Irish potato famine, was the result of this one pathogen getting introduced and destroying the potato crops. And just a really grim diagram here. This is percent population decrease, 30% decrease in the size of the population during this period. Rather brief period, 7 years. This was also related to immigration to the United States at that time. So immigration from Ireland was rather big at this time. So kind of interesting, I guess, if you trace back the connection to plants and the pests of plants, just these world events really driven by kind of small movers, right? These microscopic pathogenic organisms. So kind of a cautionary tale for depending on one food as your food supply. So as I mentioned with the nematodes, just sort of other organisms that can attack your plants. So nematodes, we saw fungus, here's bacteria. Just some examples of bacteria. Getting into kind of a creepy image here of bacteria creeping up to a stoma. So one of those openings on the leaves of a plant, potentially an entry point for bacteria. And then once they get inside of the plant, they can cause trouble with all these different evidences of bacterial infection. Plants get viruses as well. There are viruses specific to plants. Turns out that viruses were originally discovered in plants. So they discovered that there was something smaller than a bacteria, and they filtered out the juice extract of a plant, and they filtered it so finely that even the bacteria couldn't get through. And they found out that those juice extracts could be given from one plant and infect another, and that was the start of figuring out that there was this really small thing, or a virus, that was able to infect the plant. So this is one of the kind of textbook viruses called tobacco mosaic virus. And so this would be a pest inside of cells of the plants that it infects. So a lot of these days you would probably use DNA evidence to confirm that you've got something like this, or certainly somebody who's more familiar, especially if you're studying a crop plant, you'd be probably familiar with what kinds of pests can attack your crop plant and how to identify them and how to protect them, prevent them, and so on. So on the subject of detection, this was something that I found interesting last year. They are trying to, this group was researching the ability to detect pests by basically like chemically smelling a plant and trying to identify or characterize the volatiles, the chemicals that the plant gives off as it's being infected or attacked by different organisms. So basically you take your plant, draw off the gas that it's producing and run it through your sensor. This is attached to a smartphone, and then it's able to tell you what's wrong with your plant. So interesting technology in the future. And then kind of coming back around a little bit, so we talked about plants with their secondary compounds that they produce a lot of those in defense, so defense against organisms that attack them. And a lot of our pests control options are derived from native molecules that plants make. So we've got a neonicotinoid you might have heard about. Sometimes they're called neonics for short. This is a biochemically derived version, so a modified version of nicotine, basically. So if you have nicotine as an effective anti-pest chemical produced by plants, a modified version of that might be more effective or work its way around insects that might be resistant to nicotine, and so on. So pyrethrin or pyrethroids, these are also related to naturally produced compounds, and so on. So the idea of these is that they affect, in a lot of cases, specifically insects and not vertebrate animals. So in theory anyway, not so harmful to people, very effective against insects. Big industry there. Another place if you're looking for jobs, right? People have a little bit of a knee-jerk reaction against pesticides, but certainly they are being used, whether we like them or not, right? So I guess one headline I have on here is maybe reasons not to like your pesticides. Non-target effects. So pesticides that attack insects, these are blamed for killing other insects that are not plant pests. There's concern over the world's honeybee supply and other native bees potentially being affected because they visit plants. Plants are being sprayed with pesticides. And this one I found a little bit interesting. This is put out by a company that manufactures pesticides, and they were in this image trying to suggest, it's a little bit bigger than it needs to be. There we go. Trying to suggest that it's not just, it's also the presence of these mites. So here, the parasitic mite varroa. And they have these circle diagrams of how healthy the colonies of bees are, and then that relative to the local use of neonicotinoid pesticides, as well as the abundance of these mites. And a lot of times they kind of go together. But so here's an example. Scotland has poor bee colony health, low neonicotinoid use, but high pressure of the mites. Sort of a data point in their favor that says maybe it's actually these mites that are causing trouble a little bit more than the pesticide use. Here's Australia as another data point. So high colony health, high neonicotinoid use, and then low mite pressure. So a different version. These are healthy colonies that are still being sprayed with neonicotinoids, but healthy. I'd take this all with a grain of salt. This is basically put out by the people who are in favor of promoting pesticides. So that's their offer of data. I don't know. What do you think? Thoughts? Mix of both. All right. A nice measured response. Probably. Yeah, so the whole question of bee health, it's a global concern now. I don't know. I don't feel like we really landed on a single source of blame here. So maybe that's just going to be the answer. There's lots of different kinds of bees. Honeybees are native to Eurasia. People are concerned here about our local native bees. So we have bumblebees and other kinds of bees that are important pollinators, food source for animals that eat them, and so on. And just sort of a general concern. It's good to be cautious about things like this, right? Just a general concern that maybe putting too many pesticides into the world could be a problem. All right, questions on that topic? Comments? So kind of continuing our focus on molecules here, we talked about secondary compounds. Kind of a specific category of secondary compounds are the hormone molecules. So hormones are responsible, as we'll see, for a few different processes in plants, including, as shown here, ripening. So during the process of ripening, there's a signal. So these developing fruits on the banana plant get a signal, or maybe generate a signal that they should be ripening. And the process of ripening has them change color, change the sweetness of the fruit, maybe produce a smell, and so on. So kind of connecting back to secondary compounds, this is an article about, I mentioned before with the secondary compound topic about molecules that can find their way into honey. So mad honey here is the result of a toxic compound in the flowers of this plant. And apparently you can sell it for a bunch of money, $60 a pound. Another secondary compound of interest, I guess the cinnamon challenge has died down. What year was that? Hopefully nobody tried that, right? Remember the cinnamon challenge? Take a whole bunch of cinnamon, put it in your mouth, and invariably have a coughing fit. That's it. So full of nasty chemicals. So we like cinnamon in small doses, gives a nice smell and flavor to things, but we're not meant to eat it in spoonfuls. But biologically, of course, the plant has not set itself up to harm people in the cinnamon challenge, but it is pretty effective in what's shown here in treating or inhibiting or killing different kinds of bacteria. So a little bit of a throwback, if you've taken the genetics lab, we do one of these with hydrogen peroxide. You put the chemical inside of a paper disc and then look at how well it inhibits the growth around it. So these are different concentrations of cinnamaldehyde. And this is one of the active chemicals inside of cinnamon. So it's not just a cute thing for us to put onto food, but it actually kills bacteria. So putting that into your food might have a double benefit. We think it tastes good. It also might be killing if you've got bacteria on there that could cause your tummy some trouble a little bit later. Some of these are pretty effective at killing bacteria. Okay, so on to hormones. Hormones are chemical signals, and they can cause changes in the plant kind of on different time scales. So sometimes rather quickly, sometimes altering the form of growth. So this example here, this is one hormone called Gibberellin, and this can be added to grapes to cause them to grow bigger. So as a result, this is an outside signal that's being provided to the plant during the course of development, and basically humans can take advantage of that and say, okay, we're gonna convince this plant to put more into its grapes than it would naturally. But it's taking advantage of a natural signal that might already be present at a certain amount, which is the plant signaling, okay, you're going to build some grapes using this signal called gibberellin. So as I mentioned, we talked about specialized metabolites before. Let me jump ahead here. As we saw before, the molecules that plants make, we've got this basic metabolism, and specialized metabolism builds out of that. And just showing the starting point, if you're curious about these, there's a lot of molecule diagrams today. I don't really focus on anything having to do with chemistry, but I guess as much as anything, they look nice. And if you wanted to explore these processes of making these molecules, this is basically how they branch off of the basic metabolism pathway to become specific molecules. So a few different kinds: Auxins, Cytokinins, abscisates, Gibberellins, basically produced from this pyruvate, really generic general molecule starting point. All right, so that was that. Let's go back to here. Diagram from I think this is from the plant physiology book. Probably a little more complicated than it needs to be, but basically there's some sort of response, and the presence of the hormone molecule causes the response to happen on a sort of a molecular basis. So a particular hormone is detected by a receptor molecule, and that receptor molecule passes on a signal that eventually results in an outcome. I think I'm talking about things in a little bit different order than I have the slides. So here's an example, another Gibberellin. So this GA here, this is gibberellic acid, and this is one of the hormones. Again, if you like thinking about cells, maybe this is helpful to think about. If you don't want to think about cells, you could try to focus on the take home here. I think sometimes it's just helpful to know that there's a lot going on, even if we're not understanding all the steps that are going on. So gibberellic acid present in the environment. So as a hormone, this could be in high amounts, low amounts. And based on the amounts of gibberellic acid, that causes more or less of it to interact with these receptor molecules. And then, basically, we're not going to follow the points on here, but you can follow the arrows around. And these arrows go, as we go through this pathway, to various parts, turn on transcription of genes, cause vesicle formation and molecules to be made. This alpha amylase molecule is a starch digesting molecule. And so that's all being produced and then exported, and this diagram is showing a process happening in seed cells. So gibberellic acid, one of the outcomes in this organism is to break down the starch in the endosperm. If you remember, way back when we did seed germination, some of the seeds we tried to germinate, didn't germinate many successfully, but we tried adding this gibberellic acid, which is involved in causing seed germination. So germinating seeds, one of the things that gets clicked on in that process is to break down the endosperm starch. What happens when you break down starch? What do you get? Sugar, right. So starch is a good energy storage molecule, longer term energy storage, but sugar is more immediately available. So breaking down the starch into sugar makes that available for the embryo. And as part of seed germination, that embryo is going to need some energy to get going. So you kind of see the signal and the results all in this one diagram. So gibberellins are one class of hormones, as are a bunch of these other ones. Seems like more keep getting discovered. I have a little bit of a hard time presenting about hormones because there's a lot of specific interactions, and it's a little hard sometimes to generalize about, yeah, that's definitely the molecule that's involved in X or Y, because there are just so many molecules present, and sometimes we have competing signals from different sources. We have different cells responding differently to the same hormone. We have different organisms, different species that might respond differently. So I'll try to give you kind of a general impression and an introduction, at least, to these different hormone categories. But the specifics of any one of these can be a lot to wade through as you're learning about them. Does ethylene make bananas brown? Basically. Yeah, so we'll get to that one later. Yeah, ethylene is one of our hormones involved in ripening. Good one. And then also, so we've seen a little bit about gibberellins. We'll also talk about Auxin, cytokinins, abscisates. Not as much on jasminates here. Okay. Right. So here's one. As I mentioned, it's a little hard to generalize. Here's one of the reasons it's a little harder to generalize about hormones is that So it's sort of a molecular signal, and it can be a specific molecular signal. So different parts of the plant are getting different hormone signals, and they might be responding differently to the same signals. So here's an illustration of different cells. You might have a plant that's experiencing an abundance of ethylene. So let's say you put it in a bag with your ripening bananas. Different parts of that plant might be responding differently. So as I looked into the functions of ethylene, causes leaf cells to die, seed cells to germinate, and so on, ripening, flowering. So the signal is sometimes produced kind of locally, or it might be produced more throughout the plant more evenly, but the responses are different depending on the cells that receive the signal. So we're kind of getting into physiology, biochemistry. So I try to kind of gloss over it a little bit without getting too deep into the concepts. In fact, I'm going to probably just skip right through this one, but so we have classes of hormones and then known effects and also locations where these hormones are produced a little bit more often. Maybe it's worth dwelling on this slide a little bit, just give you an idea of the kinds of processes that are influenced by hormones. So leaf senescence, so leaves dying at the end of the summer, maybe as the plant gets older, can be encouraged or delayed. Leaf senescence stimulates cell division, stimulates root meristem formation, and so on. So lots of different research directions to take on this one. This is a study that was trying to synthesize a lot of the information about where these hormones are found in the plant. So shoot apex, root apex, getting in specifically like just the apex of the shoot, and then more specifically single cells within there, or even organelles within different parts of the plant. So these are colored by reference sources on this article, and also colored by the types of hormones involved. So shoot apex, a whole bunch of the cytokinin examples, a little bit of Auxin, Gibberellins, and so on. Okay, so what kind of signals does a plant need as it goes through its life? We saw a little bit already. We talked about talked about ripening fruit, right? So we take a fruit that's being developed. There's cells in there. Green, say we got a green banana, the plant is making on the pathway to develop a banana. The cells then later get a signal that says, all right, go through this process we call ripening, of changing color and sugar content and so on. And that process can be encouraged by hormones. A lot of these have to do with growth. We saw the process of switching from vegetative to reproductive. So I guess I want us to think about this in terms of cells, right? A plant cell is just like a little isolated cube somewhere in the plant, and it gets a signal about what to do based on its environment, right? So within the plant, there can still be signals coming from other parts of the plant that say, you need to grow bigger. You need to, sometimes they get the signal to die, right? We're done with you, cell. Produce certain molecules, so on. Let's see. We'll put one in there. So produce. Let's say there's a plant that needs to set up to defend itself. Ripening on there again. So a single cell getting a signal, single cell getting a signal to do something for itself. So I chose this image just to kind of show different parts in the life of a flower, right? So the plant got the signal to make flowers in the first place. That might be driven, as we saw way back, we talked about the initiation of reproductive growth. Could be signaled by something like day length, plant size. Some of that signal could be involving hormones as well. Produces a plant, the flower opens. I'm sorry, produces a flower, the flower opens. A little bit different signal. This flower then switches gears. The petals fall off. The stamens shrivel up. The ovaries develop. Seeds develop. So as far as individual cells, they might need to know and get their marching orders at different times for what they should be doing. Are they part of the plant that's going to grow, part of a plant that's going to fall off? So during the course of development, in response to the environment. So some of these hormone molecules are produced in response to being chewed upon, for example, or maybe conditions being colder, sunnier. We saw the production of colors in leaves, right? So some of those leaf colors are produced in the fall, some of those anthocyanins. There's the end of the season growth when the chlorophyll is brought in from the leaves. So again, signals involving that process. They don't have to be every cell making its own decision. They can kind of get these decisions passed on to them by other cells using hormones. Okay, so I think we maybe covered this types of response. So cells that divide, grow. I mentioned cell death is an option. Seed germination as suggested here. So one of the first hormones, probably one of the most commonly talked about hormones, is in this group called the Auxins. And Auxin actually gets its name from the idea of growth. So if you want to impress your friends, you could use a word like augment, right? So augmented, this shows up in augmented reality. Is that a thing still? You put on your special glasses and have Pokemon show up in the park. That's augmented reality. So augment, to increase or add on to. That's the root of this word auxin, basically the same root. So increasing in size. Here's an illustration from the model organism Arabidopsis that shows if you have mutants in their ability to sense auxin, this mutant here is having a really hard time growing. It's not getting the signal. So the auxin signal is being produced by the plant, but the molecules that are supposed to respond to that aren't getting the signal, and that causes this whole plant to stay a lot smaller. So we understand pretty well about the molecules that respond to auxin as well. This is also one of the original molecules really to be investigated. So this is all the way back to our friend Darwin, 1880, did an experiment on growing parts of plants in the grass family. And it was determined that auxin is responsible for causing this seedling shoot to bend toward the light, right? So if you have light off to one side of the plant, then it will, the plant will respond by moving in that direction. So that's been known for a while, right? You can grow your plants in the window and you'll notice that kind of bend towards the light. Quite a few different kinds of plants do that. The investigation was, okay, well, what causes that to happen? So a few different examples here. This one had them cutting off the shoot tip, and in the absence of the shoot tip, there was no response to light. So that told them that there was something in there that was important to sense the presence of light and respond to it. Here's another one where they covered the tip with a little a cap that prevented light from getting in, and that prevented this shoot from bending over to the side. So that was important. These other parts, the more stem part of this shoot, we're still able to get the sun signal from one side, but the absence of the signal in the tip of the plant prevented it from bending. Here's one that's kind of a control for, all right, maybe it was being affected because we, prevented air from getting in there or had contact on the shoot tip. Maybe that was affecting it. So they put a clear helmet on this one, and that was still able to sense the light and bend toward it. And in this example here, they prevented the light from getting to the stem cells, and it was still able to bend. The part of this plant that was able to bend still did that. So basically, the take-home from this is that there's something in the shoot tip that's really directing this whole plant growth off to the side.
Plant hormones, also known as plant growth factors, are chemical substances that accelerate, inhibit, or otherwise affect growth. This animation explores the action of indole-3-acetic acid, IAA, the most common form of auxin, and looks at how we've discovered what auxins do. The auxin IAA is produced at the apex of the stem. Under unilateral light, shining on one side, IAA is redistributed to the shaded side, creating a gradient of the hormone across the stem. IAA travels between cells by diffusion. IAA activates proton pumps, which lowers the pH. This activates enzymes, expansins, to break bonds in the cell wall. The loosening of the cell wall allows water to enter the cell and turgor pressure increases. Cells on the shaded side become elongated and cause the shoot tips to bend towards the light. But what is the experimental evidence for this? Darwin investigated using oat coleoptiles. He found that there is no bending response to light when the coleoptile tip is removed. This means that either the tip is the origin of the messenger, or it must be stimulated by light. To investigate further, Darwin covered the tip to prevent light from reaching it. The lack of bending response confirmed that the tip must be stimulated. When light was shone on the tip but the lower regions were covered, the coleoptile did bend. This showed that only the tip needs to be stimulated. Peter Boysen-Jensen used a thin flake of the mineral mica, which blocks chemical movement. When it was inserted in the illuminated side, bending occurred normally. But when the mica was inserted in the shaded side, there was no bending, showing that the message to bend travels down the shaded side. Because the mica doesn't block electrical signals, this also suggested that the message was chemical. Arpad Pal's experiment replacing a segment of the tip off-center, increased understanding of how the hormone is distributed. Even in the dark, the coleoptile bent towards the side without the tip. Fritz Wendt removed the coleoptile tip and incubated it on a block of agar. The agar was then placed on the coleoptile off-center in darkness. Hormone that the agar had absorbed travelled down the side of the shoot, causing a response. The amount of bending is proportional to the amount of hormone in the agar. Winslow Briggs inserted a very thin glass plate vertically through the tip. The glass does not prevent light transmission, but there was no bending. Auxin cannot travel through glass, and its concentrations remained equal on either side. When the glass plate was inserted below the tip, Auxin moved across to establish a hormone gradient. When the tip was examined, 70% of Auxin was found on the shaded side.
All right, so as I mentioned, basically one of the best known, best studied hormones, the auxins. So there's a group of these called auxin. They're related molecules, as you can see, with little chemical differences. So IAA is kind of the most famous auxin. Other ones also, IBA, IPA, and so on. I found myself, I guess, getting a lot of these studies would have a lot of chemicals and molecules in them. So as the video suggested, the shoot tip is responsible for manufacturing auxin, and then where that auxin gets transported to can give you different responses. So on the left here, this illustrates that auxin is a derivative not too far off from a common amino acid, tryptophan. And then these are molecules, the pin molecules are one of the ones that are responsible. These are transport proteins responsible for transporting the auxin hormone in one direction, kind of away from one place toward other places in shoot tip. So making it is important, as is getting it around in the cell, in between different cells. Let me skip that one. It's not really my cup of tea, but there's certainly a lot of people researching these fine details of where molecules are going and how they get around in the cell. So root tip, shoot tip, transport of auxin dependent on a lot of these different molecules. So this pin family of proteins, OX protein, and so on. So it's a lot going on. We saw before, we talked about phyllotaxis when we talked about the positions of leaves along the stem. And the illustration that I showed you something very similar to this much before, identified that some cells are basically getting the signal to turn into leaves. And the pattern of where those cells get their signal, that determines the pattern of how those leaves emerge along the stem. So you get this spiral pattern as is being shown here, so offset by about 137 degrees, one leaf to the next. That's partly determined by the movement of auxin into those developing leaves. So this is more of a schematic, and then this one is showing the receptors, the transporters of where this auxin is getting moved to. Those are modified here to light up under the investigation. And there's a little bit of a difference in where those are getting put. So the shoot meristem is like a dome of cells, but the auxin is put disproportionately into different parts of that dome, and those parts then determine where the leaves are made. So kind of important. As I mentioned, Auxin made by the shoot tip. It encourages growth at the tip. It also has an interesting side function in this feature called apical dominance that we also talked about before, where side buds, these lateral axillary buds close to the tip of a shoot stem, the tip of a shoot, sorry, they're prevented from growing. So they get this, basically a signal from the auxin actually prevents growth in this location. And as the auxin concentration goes down, that allows for the growth to happen here. Also happens if you cut the tip of a plant. So if you destroy the apical meristem, that cuts away the auxin signal and allows these side shoots to grow. And I think I mentioned before, this is a process that Auxin has been credited for a whole bunch. And then this study was highlighting how you could also get a similar response basically just by looking at the sugars. So this production of sugar and making sugar available to the stem, to the shoot tip. If the tip is not using that sugar to make new cells and make new organs and so on, then the sugar gets rerouted to something near the tip, and they were claiming that that has a big role in also promoting this side growth. So a little bit of auxin, a little bit of sugar responsible for this growth of the lateral shoots. One of the ways, the video we were watching mentioned this one, one of the ways that auxin promotes cell growth is really just making it easier for cells to grow. So plant cells have a variety of firmness. We talk about the cell wall. They usually get depicted as this rigid rectangle of cellular material. But as we see, they're like wrapped up in cellulose. And as they start off initially, they are able to grow and expand quite a bit because they're not wrapped up in quite as much cellulose. And so auxin can change the rigidity of the cell wall. So shown here, some signal from the auxin causes these cellulose connections to relax. So molecules we saw before, the pectins and the hemicelluloses, those can be added to make the cell wall more rigid. They can be removed to make the cell wall more flexible. And so auxin basically loosens up all of that cell wall construction and allows the cell to get bigger. So I'm going to go over to the video was nice in highlighting that feature of causing the plant to grow towards the light. So the auxin is made at the tip. If the light is coming in from one side, then the auxin goes down the other side, the dark side. And so what that causes these cells to be more able to expand, and these cells expand a little bit less. And so if you take a growing cylinder and you grow one side more than the other, then it ends up basically pushing over. So I always found that kind of interesting. To grow towards the light is actually achieved by growing more from the dark side. So the dark side kind of pushes and causes this whole thing to move over a little bit more. So this relates to what we were just looking at with the function of auxin allowing these cells to get bigger. So elongating, not necessarily dividing to make more cells, but making those cells longer on the dark side of the shoot. Another function for auxin is in ripening. So the auxin signal for ripening is, as it says here, signal from the seeds. So plants that are getting pollinated, getting fertilized, they might not always have successful fertilization, right? So if you have a flower and it ripens, or let's say it fertilizes 2 out of 100 seeds, those seeds might not be enough to signal fruit development. And that kind of makes sense, right? So the fruit is this nutrient bundle that protects the seeds, maybe causes an animal to disperse them, and making a fruit is expensive. So the plants can control whether they make the fruit based on how many viable seeds are inside. So a small number of seeds might not be enough to tip the scales to make the fruit. A large number of seeds means that the plant finds it worthwhile to make the fruit around those. So the seeds say, hey, I'm a fertilized seed. I'm ready to go. I need a fruit. And so they're kind of signaling using these hormones to signal the plant to invest in the fruit around them. And we know this because you can take a plant and kind of artificially cause ripening to happen, even if you're not getting the signal. So you can apply Auxin kind of to stand in for that seed signal. Or there are mutants that we know about that a lot of times if you have a mutation, the ones that we find interesting to study in this context are ones that make cells not respond to the hormone signals in the usual way. So on the right-hand side here, these two are tomatoes that are ripening with no seeds on the inside. So no fertilization happened, and this plant still invested in making a fruit. So not in its best interest, but very interesting for us to know about in the lab. And maybe you could see a potential application here, right? So the seedless tomato is a plant that produces tomatoes even when there are no viable seeds in there. I don't know about you. When I eat a tomato, I don't really care if those seeds are viable, right? So our interest when we cultivate and grow tomatoes, we're interested in fruit flesh. The plant in natural conditions is interested in getting seeds around in the world. So it sort of serves us a lot better, these mutated versions that are making fruits in the absence of seeds. And a little more extreme example shown on the left here. It's a natural banana and a cultivated banana. So the bananas we eat, if you know where to look, you can find little black specks in there, which are like shriveled up, failed seeds. And again, from the plant's perspective, that's not great. If that plant wanted to make seeds to make a new banana plant, it would be out of luck. From our perspective, you look at these and ask yourself which banana you'd rather eat. So this banana, we'll talk about this again when we talk about cultivation. This is one of the changes that often happens when people domesticate a plant, is that it is investing a lot more in the flesh, which is interesting to animals, and investing a lot less in the seed, which is really the plant's objective. Are the bananas we eat genetically modified? Good question. So we'll talk about that when we talk about the genetic modification topic. Let me just double check here. That one is, yeah, that's like our last scheduled lecture, but I'll talk about it a little bit here. Genetic modification it's a loaded concept right now. So when you say that, some people have a knee-jerk reaction and they say, no genetic modification. Right, meaning only things that are natural. Other people on the other end will say, no problem, go ahead and modify my food. I trust you to test that it's not going to kill me. And so we're not always using the same language to talk about genetic modification. To me, I think when we talk about genetically modified, I like to stick with a more laboratory definition for that. So if people are manipulating the genes in the laboratory, deleting genes or something like that, I don't know where the official scientific line is on this one. That's more what I think of for genetic modification. A lot of people like to point out that most of what we eat, most plants that we eat, are modified somehow. Big difference in the genetic modification conversation is that the modifications that existed 100 years ago, those were achieved by breeding, by random, kind of more natural mutations. And I guess, I don't know, they're a little bit more natural than some of the more extreme genetic modifications. You can take like genes from bacteria and put them into a plant and cause the plant to make bacterial proteins. That's a little more extreme genetic modification. So our bananas are genetically modified, basically natural mutants that people found were much more something they wanted to cultivate. So we got a week from today, we're scheduled to talk about cultivation, domestication, these processes that happen when people take plants and grow them. They select for features of those plants that they find more interesting, more useful. So they're modified from their natural relatives, the bananas are. They're not so extremely modified that they've been like modified in the lab. I don't know if that helps. Yeah, just about everything we eat is modified. Plants don't make fruits as big as the ones that we usually eat. All right, here we go. Lab GMO. All right, there we go. Thank you, Tom. I think that's the line I'm trying to draw, right? So a potato is different from a wild potato because people cultivated it over 1,000 years. Genetically modified plants that make their own pesticides, those are something we've done only in the last generation. And there are not, as Tom points out, not too many of those in our rotation right now. So some of the big players in crops are genetically modified in the lab. Other ones are not so modified. All right. But I'm glad you're excited about the topic. We'll get another chance to talk about that. Okay. back on schedule. There is a synthetic auxin that people use as a herbicide. So this is a herbicide called 2,4-D. And it's something you can spray on your plants and causes them to basically to grow out of control to the point where they exhaust their resources. So see a plant that's been sprayed with 2,4-D, kind of takes on a contorted look. The growth is not so organized anymore. So this is like individual cells growing at their own paces. Gives you a, like I said, kind of a contorted look to the whole plant. So the auxin we mostly talked about is that IAA form. Another type of auxin is IBA. This is sold as a rooting hormone. I don't know if anybody's used rooting hormone before. So you can use rooting hormones on certain plants to cut them, and you take a piece of the stem, and the IBA hormone encourages the stems, the ground tissue, the parenchyma cells in the stem, encourages them to make root tissue. So nice use where we can kind of co-opt this natural signal and tell stem tissue to make root tissue through an application of so-called rooting hormone. And you can see it's just the whole of this rooting hormone, that's the active ingredient, and also a very small amount. So hormones are also very effective at small doses. Yeah, didn't see a noticeable difference. You know, I haven't used it much myself. I think it might depend on the plant. Going back to Kennedy's questions, Miracle Grow, as far as I know, Miracle Grow is only nutrients. I don't believe they add hormones to fertilizer. Hormones are kind of touchy. You know, if you get the wrong combination or maybe the wrong part of the plant or species of plant, you could be signaling something rather disastrous. So, you know, for example, here, this is a hormone that encourages growth, but in like just a blanket application on the leaves of a plant, this causes really the wrong kind of growth. So if you want to get a hormone, you basically will buy just the hormone. Causing your plants to grow well with a fertilizer, that's a whole different thing. That just basically is providing them with more nutrients that allow them to build more structures. Now the connection, I guess the connection between nutrients and hormones, there might be a connection there where the plant is sensing that it has more nutrient availability and then shifting gears on how it grows as a result of that. Whole wide open topic today. Another type of molecule, so that was a lot about auxins. This is another type called cytokinins. They're named for the process of cytokinesis or cell division. And here's a couple of them, zeatin and kinetin. And one of the big sources, as you can see here, the big source of cytokinin is coconut milk. So if you want to get a cytokinin effect on your plant, researchers would, before we had maybe as many purified chemicals at our disposal, people could just take the milk of a coconut and use that as their cytokinin signal. I don't know why I have all these on here. This is a kind of a basic starting point. These are ATP, AMP. These are nucleotide and ATP, this energy-containing molecule, and then through some conversion steps, that's how you get over here to the zeatin molecules. Showing similarly to auxin, so we saw auxin produced at the shoot tip and transported from there. This is cytokinin produced at the root tip and transported from there. So I guess maybe if you wanted to generalize about your molecule, cytokinins more produced from the roots, transported upwards. So kind of sets up in the plant this gradient. So an individual cell is getting a little bit of auxin signal from the top and some cytokinin signal from below. Would help these cells to know where they stand in the span of the whole plant. Cytokinins, so this name cytokinin related to cell division or cytokinesis. It's a molecule that can be used to encourage growth. I showed this before, I think, when we talked about tissue culture. So you can take little bits of plant and give them hormone signals that cause them to develop into whole new plants. These are bits of tobacco leaves that are growing, just kind of a little bit of uncontrolled growth, and then at parts on here, organized growth and producing some leaves. So over time, you could grow these into whole new tobacco plants by like basically cloning cells out of a leaf. So auxin, cytokinin are kind of at odds. Tip of the shoot, tip of the root, and as I said, different cells getting different amounts of those. So if you wanted to grow your tissue culture plants, this is a nice illustration of the relative amounts of those. So kind of the sweet spot of auxin mixed with cytokinin causes the best growth kind of in this middle zone here at low cytokinin, middle auxin amounts. but also just kind of illustrates the kind of a fine-tuning of this signal. So individual cells, if you want them to grow a certain way and you're doing this in a research lab, then you really got to find your balance just right. All right. Maybe have a little bit of time for this one. Oh, we don't have time. Let's stop there. I don't know where the time got away from us. We're not too far from the end. We talked about some of the big hormones. We'll get to other ones later. So, any questions from today? All right, then I will see you on Thursday, hopefully with some grades. Let me know if you have any questions as you're doing your research project. And have a good day.